Let's look at smooth muscle. The book, for whatever reason, decides to gloss over a little bit of cardiac and go deep into smooth. I will star things that I would like you to know. All right. Those would be the things that I will probably ask more. If it's not starred, that doesn't mean it's not important. It's just that I'm not going to ask about it on this exam. Probably geared to more in the next one. Okay. When we see this again. Smooth muscle. This is your muscle of organs. Okay. Viscera, for the most part. Stuff that is, in organs automatically just happening. It's smooth muscle. It's involuntary, right? You don't control it. Blood vessels of the cardiovascular system, the bronchioles of the respiratory system, the intestines of the digestive system, ureters, the uterus and the female. We'll talk about smooth muscle in all of these various different regions. We'll talk about it more when we talk about really the first kind of three, more so than anything, in the cardiovascular, respiratory, and digestive systems. So what is it? It is spindle shaped. We talked about that in the very beginning when we compared the three types of muscle. Very wide in the middle, tapered at the ends. It's smaller. Okay, it is significant. I don't know. The sarcolemma on this one, where we have voltage gated sodium and potassium channels on the skeletal muscle. We also have some calcium gated channels here, and it can be very unique. When we look at the overall shape of this thing, it's really unique. It doesn't have T tubules. It doesn't have any transverse tubules. It has what's called an invagination. That's like a flask, and that's called caveolae. It's a flask-like invagination. That's where electricity travels down in and affects the actin and myosin inside. Very sparse sarcoplasmic reticulum, and that's similar in retrospect to the cardiac muscle, because the heart really never relaxes. It doesn't have an extensive sarcoplasmic reticulum or have cisternae even, for that matter. Kind of same mantra here within the smooth muscle as well. It relies more so on calcium being pumped into the smooth muscle itself, not like skeletal muscle. So here you can kind of see it. Looks very weird. Remember, no sarcomeres here, hence smooth muscle name, and that I believe is on the next slide. But you can see we have kind of this weird arrangement of thick and thin filaments and some intermediate filaments, and even these things called dense bodies that seem to hold everything together. They're like little beads that hold everything in place. So a very fantastical structure when you look at it. Intermediate filaments. Intermediate filaments are there to kind of help anchor the actin and myosin, the contractile proteins. Those are the ones that we're most concerned about, to be honest. Oh, I said I was going to highlight stuff, right? It's smooth muscle. Give me an example of organs. It's stuff that you don't control. It's fusiform shape. T tubules absent. Sarcoplasmic reticulum sparse. Intermediate filaments are here for one thing, to help to anchor the contractile proteins down. That's what I would like you to know there. Again, they're not in a repeating fashion. So, I mean, there is no sarcomeres here, right? There's no Z disc. If there's no sarcomere, there's no Z disc, right? So it lacks all that stuff. All of the contractile proteins, the actin and myosin, still there, just not in any normal sort of arrangement, are orientated in this weird oblique, they're just at angles to each other, right? When we look at how smooth muscle contracts and relaxes, it functions like, okay, with that sound obviously as well. There are, again, like I said, actin, myosin. We have the ability to, it's called a latch bridge mechanism. And that just means I can stay attached for an extended time. Okay. I don't have to input any extra ATP to do it. It's able to grab the actin myosin are able to interact with each other and hold that contraction for an extended period of time, okay? Which is different from everything else that, we've talked about really so far. And even cardiac muscle, it contracts, it relaxes. Here it contracts and relaxes. Not quite to scale. We have calmodulin here. Instead of helping the interaction with troponin and tropomyosin, we have this stuff called calmodulin. That's what helps to kickstart the jam here. We have a number of different things called myosin light chain kinase and myosin light chain phosphatase. These are going to be the specific enzymes that interact the phosphorylation to get myosin head to go up and interact with actin. But I didn't underline those two things. We'll talk about those two later. Here's the steps of it. I'm not going to ask it as a short answer question if I was to ask this on the exam. Okay, it would only be as a multiple choice type question. But here it's pretty similar. Stimulus. Okay, we open up voltage gated calcium channels here. Calcium enters, binds to calmodulin, binds to myosin lactate kinase. You know what? I'm not even going to ask. Not even going to ask. I really want you to know everything that has to do with getting the skeletal muscle contract. Neuromuscular junction, at the motor end plate, along the sarcolemma, down the T-tubule, in the sarcolemma. I really want you to know that one, okay? So slide 86, don't worry about it, okay? I won't ask any questions about it. Here's some pictures to kind of see how it does it. Again, I'm not asking about it. Relaxation, obviously, I'm not going to ask you about it. We just stop the stimulus and really everything goes in reverse. We'll look at it a little bit later. Some characteristics here. It can go for a very long duration. Okay. Whoops. It can go for a very long duration. I mentioned that earlier. There it is again. Some other things. Because it can utilize energy pretty effectively and efficiently, I don't have to add any more ATP in to get these long duration contractions to occur. Very fatigue resistant. I mean, that would really suck if your small intestines got tired because you're eating a lot. Could you imagine the backlog that would happen with most people? Good gravy. So long duration and fatigue resistant, those two things are very common for smooth muscle. Those two, I don't think are too difficult to remember. It is involuntary, so it's under autonomic control. It has a myogenic response. It has a stress response. When it's stretched, as food enters into the tube, let's say, that stretches it out and that causes some contraction. And that helps to push the food forward through the tube. After a while, after a prolonged stretch, it will relax. So it can accommodate the material because it does take several hours to get materials to move through different regions of the digestive tract. So again, we want this thing to really be effective, be fatigue resistant. And if something fills it for a long period of time, I don't want to have to hold that contraction for 4 hours. It can relax, okay? It can relax. So I can break down the nutrients in that region. We do have a couple of different types of smooth muscle, and this I would want you to know. Single unit is the most common type of smooth muscle we have. All right. When it says single unit, the whole muscle is acting as a unit. All right. We form really sheets within an organ. All right. And here, one sheet, two sheets, and we see we have an autonomic motor neuron right through here, okay, right through here. It goes and supplies everybody, so the whole thing acts. Here you can see these little swellings along the motor axon. When we looked at skeletal muscle, it was just a synaptic knob. Well, here, that swelling is called a varicosity. Okay, it's something different between this and skeletal muscle. Synaptic knob is the swelling at the end of it. On a motor axon here, on single motor units or single unit smooth muscle, I have varicosities. So varicosities you only see with single unit. And again, this is the vast majority. This is what we find really all over for the most part in our system, except for a few specific locations. They can, and these varicosities, I mean, they have our synaptic vessels in it, so they're going to have neurotransmitters. Okay, that's all you need to worry about. Synaptic vesicles contain neurotransmitters. They contain acetylcholine. They contain this other guy called norepinephrine here. Multi-unit. Okay. When it comes to a multi-unit, again, this is going to be going to things that automatically happen. Erector pili in the muscles is, that smooth muscle. It just contracts. Iris in the ciliary muscle, and the degree to which your hairs can stand up a little bit, stand up a lot. That variation depends on the number of motor units, the number of muscle fibers that we stimulate. In the iris and the ciliary muscles of the eye, to get that very fine motor control that occurs automatically when light comes into the eyes, the iris, the pupil changes, okay, automatically. You don't do anything with that. And with that thing here, it will go in and it will synapse with each individual muscle cell. So that's very different here. So multi-unit, I have multiple synapses, and they're just called synapses here. I do not have varicosities. Okay. So again, we don't see those as much. Again, those are kind of the major areas, the only really big areas that you see them. Okay, in the eye, in the skin with the erector pili, you could see air passageways in the respiratory system, but really the first two are really the best examples that you can think of. But you can see here, these have synapses. These don't. These have a varicosity. Okay. So if I was to ask what is similar between the different types of smooth muscle, they're involuntary. What's different about the two types of smooth muscle, multi-unit or single unit? Well, multi-unit has a typical synapse. Every individual muscle fiber is supplied. With single unit, the whole muscle acts as a single unit. I have a motor axon that travels in between layers, and it supplies all of the muscle cells at the same time. They have varicosities and not necessarily a synaptic knob, because they don't necessarily synapse with every single muscle fiber. As you can see from the picture there, they just don't. They have gap junctions, and that impulse will travel across the surface of the sarcolemma, travel through the gap junction that connects the adjacent smooth muscle fibers, and travel in that way. So very different in terms from skeletal muscle. Kind of similar to cardiac muscle, but we'll dive into that later.